Without further ado, may I invite the first group to join the stage with such an interesting topic about identification of key genes and signaling pathways in juvenile idiopathic arthritis by bioinformatics analysis by Balaussa Seitkhan from Asfendiyarov, Kazakh National Medical University.
Can I start? Sorry. Okay. Good morning, distinguished jury members and participants. I'm glad to present our research work, Identification of Key Genes and Signaling Pathways in Juvenile Idiopathic Arthritis by Bioinformatic Analysis. And so let's start. Juvenile idiopathic arthritis, or JA, it's an autoimmune disease that occurs in children under 16 and causing persistent inflammatory processes, not only in the joints, but also in internal organs, eyes, and so on. And for your special attention, juvenile idiopathic arthritis, is a leading cause of childhood disability, and 40% of patients are not respond to first-line therapy, and so they lose their time to good treatment, and we need personalized therapy to decrease this time. And also, and there is why our goal was clear: to identify signaling pathways and hub genes associated with severe GIA based on bioinformatic analysis of transcriptomic data. And we analyzed transcriptomic data from Gene Expression Omnibus, which included 12 GIA patients and 13 healthy controls, and differential expressed gene analysis, DEG analysis, were identified using GO2R, and functional enrichment analysis was performed using EnrichR platform, and we constructed PPI network using string and visualized it in Cytoscape. Also, thanks to Cytoscape, we identified hub genes and visualized it in Cytoscape. We selected 2 hub genes and were visualized as box plots using IBM SPSS. And you can see the scheme. Next, it's a main part of our work. Of course, it's the results. So you can see the DEG analysis, the box plot confirms the proper normalization of the data, and we identified 300 DEGs as shown in the Venn diagram. Next, it's a UMAP analysis. UMAP analysis clearly separated 2 groups. It's a JA patient groups and the healthy control, which confirms biological significance of the data. And next, it's a volcano plot showed upregulated genes in red and downregulated genes in blue. It confirms the reliability of the data. So this is a statistical significance of the data. And next one, it's pathway analysis. To reveal their biological relevance, we carried out enrichment analysis separated for 2 groups. It's for upregulated genes and for downregulated genes separately. Yeah. And the first gene ontology analysis, you can see that upregulated genes enriched in cellular motility, also stress survival, and downregulated genes showed suppression of immune response. Next is the KEGG analysis, and in this slide you can see the that upregulated genes strongly linked to hormonal signals and platelet activation. And next is while downregulated genes is associated with metabolism pathways. Yeah. Last but not least, it's a reactome pathway. This reactome pathway pointed to the AKT survival AKT survival pathways and for upregulated genes, and then you can see the problems with catabolism of amino acids, in down-regulated genes. So finally, we constructed PPI network to see the connections between genes and proteins. And thanks for it, we can identify the hub genes, top 10 hub genes, and also visualize it in Cytoscape. You see the PPI network for hub genes. And box plots of hub genes, it's for, we selected two significant hub genes. It's the HSP19AAB1 and the HSP19AA1, which confirms, you can see the distinct differential expression for two groups. And the HSP-AB1, it's considered of isoform for chaperone proteins, and you know that chaperone proteins are essential for protein for defending against cellular stress. And HSP90AB1, it's a key gene which sustained expression level promotes fibroblast-like synovicites, which supporting inflammatory processes. And in JA, it's a key gene. Also, next is a HSP19AA1 gene. This gene, it's inducible as a form of chaperone proteins, and this supports the inflammation pathways such as NF-kappa B, and it also supports synovicitis in GAA. In conclusion, we identified hub genes that they are key regulators of immune response, and they can serve as the potential biomarkers for diagnosing and monitoring disease, and as therapeutic targets for personalized therapies. And these findings lay down the future studies. And also, we have already won a research grant in our university to work to validate our research findings with real-time PCR researches. Thank you for your attention. I'm glad to answer your questions.
Thank you for your nice presentation. And I have a question that how do you select the 300 genes? Because I saw from the Venn diagram that you show there is no difference between the number of genes in the sample and the control.
This one?
How do you select this 300 genes?
Yeah, of course, it's a DEG analysis. We provide this in the platform Gene Expression Omnibus. And for this, we use GeoTour tool. And look at this. It's not a sample. It's genes. And sample totally is 25. And the 300, it's genes. 12, it's 12, and 13, it's a sample. It's patients.
But we should see the difference because you show the EG, right? So we have to see the difference of the different number of genes that express in the sample and the control.
Sample and control. This shows the difference between two groups. 300. Maybe you can.
You know what I mean? It should see the expression of the gene number of the sample and also the patient, and then we will see the overlapping that show the DEG, which is DEG, right?
Yeah.
But we didn't see any overlapping of the gene, of these 300 genes.
Overall genes, you mean? Overall genes, it's like in the corner of the slide. And you can see the total of number of genes, on the corner. And which shown in the Venn diagram, it's a deck. It's a differential express genes for two groups. Total, it's in the right, it's in the corner. Yeah. Yeah. Okay. I'm glad to answer.
So you said that you have get the data from the transcriptomic database. So from which tissue that the data is coming from?
Yeah, it's from blood samples. Yeah, peripheral blood.
So peripheral blood.
Yeah.
And in the patient group, they are on the medication for the treatment as well. So how can you conclude that the data is based on the baseline of the disease or the disease after the control by the immunosuppressant agent?
Sorry, can you repeat?
So from your sample group of the JIA, the patients are with medication, which is methotrexate, that also cause the difference in the expression of the gene as well. So how can you conclude that is the effect of the disease or of the medication?
No, it's it means that therapy means the severe progression of the GAA, and it's not related with this. Yeah. It's means the severe progression. That's all. And yeah, thank you for your attention.